Hello, I am Professor Sims, and this video is for the Gen Bio 1, Lesson 3, Biological Chemicals and Macromolecules. This is the third of 10 lessons included in my General Biology 1 lecture course. In this lesson, we will discuss the study of and the role of cells, cell theory, components of and functions of the endomembrane system, cytoskeleton, microfilaments, intermediate filaments, microtubules, cilia, and flagella. We'll also talk about differences between prokaryotic, eukaryotic, animal, and plant cells, the roles of tight junctions, desmosomes, gap junctions, and plasmodesmata, how cells communicate with each other, and the processes of passive transport, active transport, and bulk transport in plasma membranes. Robert Hooke was a 15th century scientist who described the earliest observed microorganism, a microfungus called mucor. He also coined the term cell, having observed microscopic, box-like structures that resemble honeycomb cells using what is now known as a Hooke microscope. This was essentially a modified telescope with a convex objective lens, an eyepiece lens, and a tube or field lens. Its light source was an oil lamp, which gives you an idea of its optical performance. The Leeuwenhoek microscope was developed in the 1670s by Antony van Leeuwenhoek and consisted of two flat, thin metal plates riveted together. Sandwiched between the plates was a small biconvex lens capable of magnifications ranging from about 70 times to over 250 times actual size, depending on the lens quality. Now, Leeuwenhoek used this microscope to make the first observations of what he called animalcules, which helped to disprove the theory of spontaneous generation. Francesco Redi, known as the founder of experimental biology, further disproved spontaneous generation when he effectively showed that adult flies must be present for maggots to develop, maggots being the immature flies. Finally, Louis Pasteur conclusively disproved the theory of spontaneous generation with his famous swan neck flask experiment, which we will discuss in just a minute. This figure shows Francesco Redi's experimental setup, which consisted of raw meat held in 3 different containers. One had an open top, one was sealed with a cork top, and the last one was covered in a mesh. Now, the immature flies, the maggots, only appeared on the meat in the open container because that's the only container that allowed for the adult flies to go inside and lay their eggs on the meat. Maggots were also found on the mesh top up here because the adult flies were attracted to the smell of the rotting meat and they laid their eggs as near to it as possible, which would be up here on the top. This figure shows Louis Pasteur's famous swan neck flask experiment, and this is the one that definitively rejected the idea of spontaneous generation. It was the shape of the flask that allows air to come inside of the container, but it prevents the entry of any kind of bacterial or fungal spores. The experiment consisted of 2 parts. In the first part, the broth in the flask was boiled, and the flask was allowed to cool intact, right? So with this still attached. In the second part, the broth was boiled, and then the neck was broken off, like so. And once both flasks were cooled to room temperature, the first flask remained sterile, but the second was contaminated, right? Because the spores could get in. So the theory of spontaneous generation was replaced with cell theory in 1839.
There are 3 parts to the cell theory. 1. All organisms are composed of 1 or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. Once Leeuwenhoek had his microscope ready, he went to town, looking at anything and everything he could with them, including the gunk on his teeth. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. It is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today.
The basic tenets of cell theory are: all living organisms are composed of one or more cells. The cell is the basic unit of structure and organization in organisms, and cells arise from pre-existing cells. So what's a cell? A cell is the smallest unit of life. Most cells are so tiny that we can't see them with the naked eye, and therefore scientists use microscopes to study them. To give you a sense of cell size, a typical human red blood cell is about 8 millionths of a meter, or 8 micrometers, in diameter. A pinhead is about 2,000ths of a meter, that is 2 millimeters, in diameter. That means about 250 red blood cells could fit onto the very tip, the very head of a pin. So scientists use microscopes to study these tiny cells, and a microscope is an instrument that magnifies microscopic cells and organisms. And when a microscope is used for photography, the pictures that it takes are called micrographs. So you have here a nice little diagram of the limits of what you can see with the human eye versus different types of microscopes. Right, so the human eye, it goes up to about the thickness of a hair. And then after that point, you're going to need a microscope of some kind. So here we have a light microscope and electron microscope. A compound light microscope allows you to see things as small as a single cell. And an electron microscope allows you to see things much smaller, things like viruses and macromolecules, and even a single atom with a very sophisticated electron microscope. And here you can see the relative sizes of animal cells, plant cells, and bacterial cells. In general, the prokaryotic cells are going to be much smaller than eukaryotic animal and plant cells, but they're still, prokaryotic bacterial cells are still much, much larger than viruses. Viruses are 3 orders of magnitude smaller than prokaryotic bacterial cells. So that's why you need an electron microscope to view a virus. In a light microscope, visible light passes and bends through multiple lenses to enable the user to see the specimen. Light microscopes generally magnify up to 400 or 1,000 times actual size. Magnification is the process of enlarging an object in appearance. This is not the same as resolution, which is the microscope's ability to distinguish 2 adjacent structures as being 2 separate structures. In other words, the higher the resolution, the better the image's clarity and detail will be. So this is how well it focuses. In order to gain an even better understanding of cellular structure and function, scientists typically use electron microscopes. So this over here is an example of an electron microscope. You see how it's much, much larger than a light microscope. Electron microscopes provide a much higher magnification, up to about 100,000 times actual size, and they have a much higher resolution power than light microscopes as well. As you can see in the figure, electron microscopes, again, they're really large, and they use both lenses and electromagnets to visualize the specimen. And yeah, they're really sophisticated and also very expensive. Cells fall into one of two broad categories. We already talked briefly about prokaryotic and eukaryotic cells. We classify the predominantly single-celled bacteria and archaea as prokaryotes. Animal cells, plants, fungi, and protists are all eukaryotes. So what does that mean? Well, prokaryotes are single-celled with a plasma membrane. They have cytoplasm, ribosomes, and peptidoglycan cell walls. Their DNA is not membrane-bound, and they have no nuclei and no organelles. They're really quite small in size, ranging from 0.1 to about 5 micrometers in diameter. Their cell size is limited because as a cell size increases, its surface area to volume ratio decreases. And if the cell grows too large, the plasma membrane won't have enough surface area to support the rate of diffusion needed to sustain life. And since prokaryotic cells lack a membrane-bound nucleus, their genetic material is contained in a nucleoid. This figure here shows the generalized structure of a prokaryotic cell. Here you can see the nucleoid, ribosomes, a cell membrane, and a cell wall. The other structures, for example, pili and flagellum, are present in some but not all prokaryotic cells. The small size and high surface area to volume ratio of prokaryotic cells aids in nutrient and waste exchange. So as a cell increases in size, again, its surface area to volume ratio decreases, and when there's insufficient surface area to support the cell's increasing volume, a cell will either divide or it will die. Some terms to know here are transport rate, growth rate, and evolutionary rate. The transport rate is the rate of the movement of raw materials into and waste out of a cell. Growth rate is how fast a cell grows, which is limited by the transport rate. High growth rate results in increased metabolic activity and cell reproduction. Evolutionary rate is the amount of evolutionary change over time. So, given the same amount of nutrients, small cells can have more individuals in a population than can larger cells. So, smaller cells undergo more cell division, more mutation, and therefore more evolutionary change. And in this way, prokaryotic cells have an evolutionary advantage over eukaryotic cells due to their small size. Like all cells, eukaryotic cells have a plasma membrane, cytoplasm, and ribosomes. But unlike prokaryotic cells, eukaryotes also have membrane-bound nucleus and membrane-bound organelles with compartmentalized functions. I will discuss these organelles in great detail in the following slides, but briefly here, you should know that a plasma membrane is a phospholipid bilayer embedded with proteins. The nucleolus assembles ribosomes, which perform protein synthesis. Mitochondria are involved in cellular respiration, ATP production. Peroxisomes hydrolyze fatty acids, amino acids, and toxins. And vesicles and or vacuoles are involved in storage, transport, and breaking down macromolecules. Animal cells, plants, fungi, and protists are all eukaryotes. Animal cells have centrosomes and lysosomes. Plant cells do not. Plant cells have a cell wall, chloroplasts, and central vacuoles. Animal cells do not. Otherwise, animal and plant cells all have the same organelles. So let's have a look at those now. This figure shows the major organelles of a typical animal cell. The nucleus is this purple circle here. It houses the cell's DNA and directs the synthesis of ribosomes and proteins. The nuclear envelope is a double membrane structure on the outermost portion of the nucleus, which controls the passage of ions, molecules, and RNA between the nucleus and the cytoplasm. This darker circle within the nucleus is called the nucleolus, and it aids in the synthesis of ribosomes, which are the cellular structures responsible for protein synthesis. When viewed through an electron microscope, ribosomes appear either as clusters or as single tiny dots in the cytoplasm or attached to the plasma membrane, endoplasmic reticulum, or nuclear envelope. So you can see some ribosomes surrounding the nucleus and the nucleoid. And the ribosomes receive their orders for protein synthesis from the nucleus, where the DNA transcribes into messenger RNA, mRNA. And because proteins are essential for life, there are ribosomes in practically every cell. Peroxisomes, these guys here, are small round organelles that carry out oxidation reactions that break down fatty acids and amino acids. They also detoxify many poisons that can enter the body. For example, peroxisomes in liver cells detoxify alcohol. Endoplasmic reticulum is the ER, and it's connected to the nuclear envelope, which is then connected to, obviously, the nucleus and the nucleoid, right? So the ER is a network of membranous tubules, which serves multiple functions important in the synthesis, folding, and modification, and also the transport of different proteins. This here is a vacuole. Vacuoles are membrane-bound sacs that function in storage and transport. You may know the mitochondria as the powerhouse of the cell. This is because they're responsible for making adenosine triphosphate, ATP, which is the cell's main energy-carrying molecule via cellular respiration. The cytoplasm, so everything in between the different organelles, is the cytoplasm. This is the region between the plasma membrane and the nuclear envelope, and it consists of about 70% water. The other 30% is made up of proteins, sugars, amino acids, nucleic acids, fatty acids, and ions such as sodium, potassium, and calcium. Many metabolic reactions, including protein synthesis, take place in the cytoplasm. Next, we have the Golgi apparatus. This is a complex of vesicles and folded membranes responsible for transporting, modifying, and packaging proteins and lipids. Then here we have the lysosome. These are the cell's garbage disposals. Enzymes within the lysosomes aid in breaking down proteins, polysaccharides, lipids, nucleic acids, and even sometimes worn out organelles. Then, of course, the plasma membrane. This is also called the cell membrane. It's found in all cells, and it separates the interior of the cell from the outside environment. We'll talk about microfilaments, intermediate filaments, and microtubules in a little while. Also, the cytoskeleton we'll speak some more about. But I did want to discuss quickly the centrosome. This is a microtubule organizing organelle. It contains centrioles, which are important in cell division. So when we talk about mitosis, the centrioles come from the centrosome. And again, we'll talk about these other structures in just a moment, but I want to move on to plant cells first. This figure shows the organelles and components of a typical plant cell. Remember that plant cells do not have centrosomes or lysosomes, but they do have a cell wall, chloroplast, and central vacuoles. So here you can see a structure that is external to the plasma membrane. This is the cell wall. It's a rigid covering that protects the cell. It provides structural support and gives shape to the cell. While prokaryotic cell wall's chief component is peptidoglycan, the major organic molecule in the plant and some protist cell walls is cellulose. Cellulose is a polysaccharide that's comprised of glucose, and it really helps providing structure to the cell. Here, you can see a large organelle that occupies most of the cell's area. This is the central vacuole, and this plays a key role in regulating the cell's concentration of water. It gives the plant structural support as well. It also supports the cell's expansion. That is, if the central vacuole holds more water, then the cell becomes larger without having to invest energy in synthesizing new cytoplasm. Here you have chloroplasts. Chloroplasts carry out photosynthesis, which is a series of reactions that uses carbon dioxide, water, and light energy to make glucose and oxygen. This is a major difference between plants and animals. Plants are autotrophs. They can make their own food, while animals most of them are going to be heterotrophs that must ingest their food. Like mitochondria, chloroplasts have outer and inner membranes, but within the chloroplast's inner membrane is a set of stacked fluid-filled sacs called thylakoids. Each thylakoid stack is called a granum, and the fluid inside is called the stroma. The chloroplasts contain a green pigment called chlorophyll, which captures the light energy that drives the reactions of photosynthesis. Like plant cells, photosynthetic protists also have chloroplasts. Some bacteria perform photosynthesis, but their chlorophyll is not regulated to an organelle, so the chlorophyll is not necessarily inside of a chloroplast. In eukaryotes, the nuclear envelope, lysosomes, vesicles, endoplasmic reticulum, Golgi apparatus, and plasma membrane all work together to modify, package, tag, and transport proteins and lipids. And this whole system is called the endomembrane system. So let's discuss the major roles of each part of the endomembrane system now. First, the endoplasmic reticulum is made up of two parts, the rough ER, and that is going to be the modification of proteins and synthesizing phospholipids. And then the smooth ER, that's for synthesizing carbohydrates, lipids, and steroids. And the smooth endoplasmic reticulum also engages in the detoxification of medications and poisons, and it also stores some calcium ions. Golgi apparatus sorts, tags, packages, and distributes lipids and proteins. Your lysosomes are going to digest macromolecules. It recycles organelles and it destroys some pathogens. The Golgi apparatus in this white blood cell is visible here, and it looks like a stack of semicircular flattened rings in the lower portion of the image here. So it goes all the way from here to here. And you can see several vesicles near the Golgi apparatus. See all of these little vesicles? Let's look at figure 4.18. This figure shows a protein that's being synthesized and modified and then transported via the endomembrane system. So this illustration is showing an attachment of a carbohydrate in the ER here. So here's a carbohydrate and it gets attached to an endomembrane protein. Vesicles with the integral protein bud from the ER and then fuse with the Golgi apparatus's cis face. As the protein passes along the Golgi cisternae, the addition of more carbohydrates further modifies the protein. And then after synthesis is complete, it exits as an integral membrane protein of the vesicle that buds from the Golgi's transface. When the vesicle fuses with the cell membrane here, the protein becomes an integral portion of that cell membrane. So it's showing a path from the ER and the ribosomes. A carbohydrate is adding to a ribosome, which modifies it. Then it leaves the ER via a vesicle, attaches to the cis face of the Golgi apparatus, and it travels through the Golgi apparatus, having more things added to it, and it gets modified even further. Then it leaves the Golgi apparatus via another vesicle that binds to the cell membrane, and then that protein becomes part of the cell membrane. This figure 421, shows phagocytosis. You'll hear me call it phagocytosis and phagocytosis. It's the same thing. But phagocytosis is when a macrophage engulfs a potentially pathogenic bacterium and it fuses with lysosomes within the cell, and the lysosomes then destroy the potential pathogen. So in this way, lysosomes are not only aiding in digestion and organelle recycling, but also in cell immunity. This one here is showing digestion of a food particle via phagocytosis with the lysosomes. But it's essentially exactly the same process. If this was instead of a food particle, it was a potential pathogen. Now let's talk about the cytoskeleton. So the cytoskeleton is not just the cytoplasma. The cytoskeleton is a complex microscopic network of interlinking protein filaments and tubules within the cytoplasm. So the cytoskeleton helps cells to maintain their shape and their internal organization. And it also provides mechanical support that aids in cell division and in some cases in cell movement.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
The three protein elements of the cytoplasm are microfilaments, intermediate filaments, and microtubules. So let's look at all three of these in greater detail now. Microfilaments have a diameter of about 7 nanometers, and they're comprised of two globular protein intertwined strands, which we call actin, which is why they are also called actin filaments. Microfilaments, aka actin filaments, they thicken the cortex around the cell's inner edge. There are microtubules in the cell's interior where they maintain their shape by resisting compressive forces, and there are intermediate filaments throughout the cell that hold the organelles in place. Several strands of fibrous proteins wound together comprise the intermediate filaments, which get their name from the fact that their size is intermediate between that of microfilaments and microtubules. Intermediate filaments are important because they anchor the nucleus and the organelles in place within the cytoplasm. Microtubules are small hollow tubes comprised of polymerized dimers of alpha-tubulin and beta-tubulin with a diameter of about 25 nanometers. So they are much larger than the microfilaments, the actin filaments, which are, remember, those were about 7 nanometers. Microtubules help the cells to resist compression. Again, they also aid in cell division, and they're involved in cell movement because they serve as tracks for motor proteins, and they're the main structural elements of centrioles, flagella, and cilia. Flagella and cilia, along with pseudopods, are all used in cell locomotion. Pseudopods are simply projections of the cytoplasm that are formed by intermediate filaments, microtubules, and microfilaments, and then contractions of the pseudopods allow the cell to kind of move, they kind of crawl, right? Cilia and microscopic hair-like structures. Cilia are microscopic hair-like structures on the cell surface, which cause currents in whatever fluid is in the surrounding area. The currents that are surrounding the cell allow the cell to propulse. It's like they're kind of swimming. And then flagella are slender, thread-like structures. They're essentially microscopic appendages that enable many protozoa, bacteria, and spermatozoa to swim. Cilia are little tiny short appendages. They're more like hairs that wiggle, right? Whereas flagella are more like a dolphin's tail. They can swim, they can move them up and down, side to side. And then really both cilia and flagella are allowing microorganisms to swim, while microbes that have pseudopods are sort of crawling or walking instead of swimming. This figure here shows the 9+2 array of microtubules that make up cilia and flagella. So this here is a transmission micrograph of a cross-section of eukaryotic flagellum. So this was taken with one of those very powerful electron microscopes. And this is showing the sliding of microtubules relative to each other. And this actually causes the flagellum to bend. And then this here, this part C, this is an illustration of Trichomonas vaginalis, which is a flagellated protozoan parasite that causes vaginitis. It's showing its flagella. And then D here, this is a paramecium, electron micrograph of a paramecium, and it's showing that it has just rows and rows and rows of cilia surrounding its surface. These paramecium are very motile and very fast swimmers. Now we will get into more detail regarding the plasma membranes, cell walls, and how cells connect to and interact with other cells. So in plant cells, this involves intercellular organelles called plasmodesmata. Plasmodesmata are coaxial membranous channels between adjacent plant cells that directly connect the cytoplasm of neighboring plant cells to each other, establishing living bridges between cells. They facilitate the intercellular movement of various cargoes ranging from small molecules such as sugars, ions, other essential nutrients, and chemicals, to larger complex molecules like proteins and different types of RNA. Structurally, an individual channel consists of the cytoplasmic sleeve that is formed between the endoplasmic reticulum and the plasma membrane, and then this here is figure 4.28 that shows the plasmodesmata between two plant cells. So this here is a plant cell, this here is a plant cell, and this is a plasmodesma that is connecting the two to each other. The extracellular matrix, also called ECM for short, is the non-cellular component present within all tissues and organs that consist of extracellular macromolecules and minerals like collagen, enzymes, glycoproteins, and hydroxyapatite. The functions of the ECM include forming essential support structure for cells, controlling communication between cells, segregating tissues, and regulating cell processes like growth, migration, and differentiation. This is a figure from the book showing the structural components of the extracellular matrix. So we've got collagen fibers, this is plasma membrane, you have integrin microfilaments in the cytoskeleton, fibronectin, and then this is a blown out illustration of the proteoglycan complex, which includes carbohydrates, proteins, and polysaccharides. Animal cells communicate via their extracellular matrices, and they're connected to each other via tight junctions, desmosomes, and gap junctions. Tight junctions, like this one here, form watertight connections between adjacent animal cells. Essentially, tight junctions are areas where the membranes of two adjacent cells join together to form a barrier. And the cell membranes are connected by strands of transmembrane proteins like claudins and occludins. A desmosome, like this one here, this is also called a macula adherens, which is Latin for adhering spot. It's a cell structure specialized for cell-to-cell adhesion, a type of junctional complex. They're localized spot-like adhesions randomly arranged on the lateral sides of plasma membranes. A desmosome forms a very strong spot weld between the cells, linking coherence and intermediate filaments that created it. And then finally here is the gap junction. Gap junctions are specialized intercellular connections between different animal cell types, and they directly connect the cytoplasm of two cells, which allows various molecules, ions, electrical impulses, other stuff like that, to directly pass from a regulated gate between the cells. So it's a protein-lined pore that allows water and small molecules to pass between adjacent animal cells. So I combined these three figures here so it's easier to see the similarities and differences between these three types of connections between animal cells, right? So again, the tight junction, these two cells are watertight connections. These two cells are completely joined together, okay? A desmosome here, this is like a spot weld between two adjacent cells. And then again, the gap junction, these are like gated channels between cells. So again, the plasma membrane is the thing that separates the internal part of the cell to the external surrounding environment. And it also controls the passage of organic molecules, ions, water, oxygen, all that kind of stuff into and out of the cell. So all of that movement in and out of the cell is controlled by the plasma membrane. And that includes waste, right? So carbon dioxide, ammonia, those kind of things, they leave the cell passing through the plasma membrane. So modern scientists refer to the plasma membrane as the fluid mosaic model because the membrane's fluid nature is due to the temperature, fatty acid tail configuration, and cholesterols embedded in the membrane. But it's called the fluid mosaic model because of the mosaic nature of all of these different proteins and carbohydrate combinations and all this stuff. They're not firmly fixed into place in the membrane. The membrane's landscape is studded with proteins which span the membrane, and some of these proteins function in cell transport. There's two broad categories of cell transport, passive and active, and then there's also what is known as bulk transport. So we're going to talk about all three of those. First, we'll talk about passive transport. Passive transport is an automatic process that doesn't require any input of energy. For example, diffusion is a form of passive transport in which particles move either into or out of a cell from an area of higher concentration to an area of lower concentration. And again, this passive process doesn't require any energy, no ATP. And this is because substances naturally diffuse from high to lower concentration areas until homeostasis is reached. And this even happens in solutions containing more than one substance, because each substance, each molecule type will diffuse according to its own concentration gradient, independent of the others. Some factors that can affect the diffusion rate are the extent of the concentration gradient, the mass, size, solubility of the molecules, system's temperature, solvent density, surface area and thickness of the plasma membrane, and of course the distance that a substance needs to travel in order for the system to reach equilibrium or homeostasis. Nonpolar or lipid-soluble molecules pass through plasma membranes more easily than polar materials, which allows for a faster diffusion rate. Also, the greater the difference in concentration, the more rapid the diffusion. The closer the distribution of material gets to equilibrium, the slower the diffusion rate. Heavier molecules move slower, therefore they diffuse slower, and the reverse is true for lighter molecules. Higher temperatures increase the energy and therefore the molecule's movement, increasing the diffusion rate. And then lower temperatures slow everything down, which decreases the diffusion rate. As density of a solvent increases, diffusion rate decreases. Molecules slow down because they have a more difficult time passing through the denser medium. And if the medium is less dense, then diffusion rate increases. Because cells primarily use diffusion to move materials within the cytoplasm, any increase in the cytoplasm's density will inhibit the movement of materials. Increased surface area increases the diffusion rate, whereas a thicker membrane is going to decrease it. And finally, the greater the distance that a substance must travel, the slower it's going to get there. So this places an upper limitation on cell size. A large spherical cell will die because nutrients or waste can't reach or leave the cell center, respectively. Therefore, cells must either be small in size, as is the case of many prokaryotes, or they must be flattened, as with many single-celled eukaryotes. So let's just take a step back here to review some key concepts. We'll talk about osmosis, tonicity, and osmolarity. Osmosis is the movement of water through a semipermeable membrane according to the concentration gradient. Right, and a concentration gradient is simply what it's called when a solute is more concentrated in one area than the other. Tonicity is what is called when an extracellular solution changes a cell's volume via osmosis. And this is directly correlated with the cell's osmolarity. Osmolarity is the concentration of a solution in parts per liter, described as hypotonic, isotonic, or hypertonic. I'm going to show you this little clip here that helps to visualize these concepts.
We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much too big to fit through the membrane, so the only particle that can move is water. Water molecules that are not stuck to a molecule of salt are far more likely to relocate. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules on the inside have much less solvent getting in the way. Water will begin rushing out of the cell. Some water will come in, but the net movement, the overall movement, of the water molecules will be outwards and the cell will shrink. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is zero. The cell neither grows nor shrinks and is much more likely to survive.
Right, so due to this process of osmosis, some materials diffuse readily through the membrane, but others, like polar molecules, can only pass through with the help of specialized proteins called channel proteins and a mechanism called facilitated transport. In facilitated transport, materials diffuse across the membrane via concentration gradient that allows the materials to diffuse into the cell without expending any kind of energy, except that they're polar and they're repelled by the cell's membrane. They're repelled by the cell membrane's hydrophobic parts. So facilitated transport proteins shield the materials that would be repelled, and that allows them to then diffuse down the concentration gradient the way they normally would if they weren't polar. Channel proteins are the transmembrane proteins. Here, this is a channel protein, and it has a hydrophilic domain that's exposed to the intracellular and extracellular fluids, and the hydrophilic channel through their core provides a route through which the polar molecules can travel, and they're not repelled by the cell membrane like they normally would be. So those are channel proteins, and those are involved in passive transport. Now, carrier proteins are different from channel proteins. Carrier proteins are involved in active transport, and that's different from passive transport because active transport requires the use of energy in the form of ATP, remember? So carrier proteins carry substances from one side of a membrane to the other. Many carrier proteins are found in the cell membrane, though they can also be found in the membranes of internal organelles like mitochondria, chloroplasts, and the nucleolus. Carrier proteins for active transport include uniporter, which carries one molecule, symporter carries two molecules in the same direction, and antiporter, those carry two molecules but in different directions. So carrier proteins are uniport, symport, or antiport, right? Figure 510 shows substances moving down their concentration gradient across the plasma membrane with the aid of carrier proteins. And it also shows that carrier proteins change shape as they move molecules across the membrane. That's going to be pretty important in just a second. Active transport is needed to move particles against their concentration gradient. That is, from an area of lower concentration to an area of higher concentration. So that's going in the opposite direction than it normally would with just diffusion, right? So a concentration gradient can be chemical due to a difference in solute concentration, but it also can be electrical due to a difference in charge across a membrane. An electrochemical gradient arises from the combined effects of both a chemical gradient and an electrical gradient. A positive ion, for example, might diffuse into a new area down its concentration gradient, but if it is diffusing into an area of net positive charge, its electrical gradient is hampering the diffusion. In this case, active transport of the positive ions occurs via integral carrier proteins in the cell membrane, which act like pumps. One of the most important pumps in animal cells is the sodium-potassium pump, which maintains the electrochemical gradient in living cells. The sodium-potassium pump moves potassium into the cell while moving sodium out of the cell at the same time and at a ratio of 3 sodium ions for every 2 potassium ions moved in. So the process goes like this. First, 3 sodium ions bind to the protein carrier. Next, the protein carrier hydrolyzes ATP and a low-energy phosphate group attaches to it. As a result, the carrier changes shape and it reorients itself towards the membrane's exterior. The protein's affinity for sodium decreases and the 3 sodium ions leave the carrier. Then the shape change increases the carrier's affinity for potassium ions and 2 such ions attach to the protein. Subsequently, the low-energy phosphate group detaches from the carrier, and with the phosphate group detached and potassium ions attached, the carrier protein repositions itself towards the cell's interior. And then finally, the carrier protein in its new configuration has a decreased affinity for potassium. The 2 ions move into the cytoplasm. The protein now has a higher affinity for sodium ions, so the whole process starts again. So this is an example of primary active transport, and it is showing how a sodium-potassium pump works. So you have 3 sodium ions here, protein changes shape, ATP, when the ATP attaches to the protein, the sodium ions are released. Then you have potassium ions that bind to the protein. A phosphate is attached to the phosphate detaches from the ATP, which makes the protein change shape again, and the potassium ions get released on this side. So that is primary active transport. Now we have secondary active transport. Secondary active transport utilizes the electrochemical gradient formed by ion pumps to move an ion down its concentration gradient at the same time as it moves another ion against its gradient. So in active transport, we have these proteins that are changing shape and they're acting as pumps, right? In secondary active transport, we have kind of like a revolving door happening. And the pumps move one thing one way and then they move another thing the other way. Whereas these secondary transports are moving things at the same time. This process is also called co-transport, and the figure is showing the co-transport of sodium down its concentration gradient and into the cell, and glucose out of the cell. And this transfer of glucose is secondary to the transport of sodium, and it's active because it requires ATP. It requires energy. Finally, let's discuss bulk transport. We touched on a little bit of this earlier when we were talking about phagocytosis. So there's two main categories of bulk transport. These are endocytosis and exocytosis. First, we're going to go over endocytosis. Endocytosis is a cellular process in which substances are brought into the cell. Three types of endocytosis include phagocytosis, pinocytosis, and receptor-mediated cytosis. In phagocytosis, other cells can engulf large particles like macromolecules, cell parts, whole cells, or like we saw earlier, pathogens. And a portion of the membrane invaginates and flows around the particle, eventually pinching off and leaving the particle entirely enclosed by the plasma membrane's envelope. Pinocytosis is similar to phagocytosis, but it's on a smaller scale. The plasma membrane invaginates and pinches off, producing a very small envelope of fluid from outside the cell, and this process is used to import substances that the cell needs from the extracellular fluid. Most of the time it's water or ions mixed in with the water. And then receptor-mediated endocytosis is a form of endocytosis in which receptor proteins on the cell surface are used to capture a specific target molecule. The receptors, which are transmembrane proteins, cluster in regions of the plasma membrane known as coated pits. So this coated pit can be seen here in figure 522. Figure 520 and 521 help illustrate the difference between phagocytosis, which is shown here, and pinocytosis, shown here. The difference between these two really is just a factor of scale. Phagocytosis is used for larger particles, and that's why it's like eating, right? And pinocytosis is used more for small ions and water, which is why it's called cell drinking. Now, exocytosis is the reverse of endocytosis. Its purpose is to expel material from the cell and into the extracellular fluid. Examples include extracellular matrix protein secretion and neurotransmitter secretion into the synaptic cleft. Now, these are very complex processes. These are complex processes that involve specific cell surface proteins, integrins, discoidin domain receptors, cell surface prostaglandins. Okay, that's just to name a few. So these are complex processes. We're not going to go into great detail on the different types of exocytosis. But for now, you just need to be sure that you know that in exocytosis, the vesicles containing the substances fuse with the plasma membrane, and then the contents of the vesicle are then released out into the cell's exterior. And here is a short clip that helps to compare and contrast the endo versus exocytosis processes.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your comments and questions for me in the comments section below.